let's begin, and I'm going to try and read off the back screen from here. And you'll see these notes on there. These are, you no need to write down what you see on the slides because the notes are simply a PDF of my slides. You can download those at your leisure, or you can download them and follow along. So that gives you a little preview up there as to what I'm going to cover. And when I saw how much time was allotted, there is put the fire hose up to your mouth, engage it, and we'll see how it's like to drink out of a fire hose. I did leave time, hopefully, for some questions. So let me begin with the first one. Is there a mind behind life? And I want to begin with a hypothesis. This is an hypothesis I've arrived at over many years of thinking, decades of thinking about this. And the hypothesis goes something like this. The mind, a unique attribute of an intelligent mind is the ability to produce significant levels of functional information. Now, there's a couple of words to note here. The word unique means that the ability to produce statistically significant levels of functional information is found nowhere else but from a mind. Now, you can generate information just through random noise. You can generate it accidentally, and you can do a statistical analysis and say, now, how likely was it that we would get the word it from this box alphabet characters we dropped on the floor? And you'll find, yeah, statistically insignificant. We did generate some information, but it's not all that important. The other word to note here is significant. There's ways in science to try and figure out if this is significant or not, and that's key in this area because, as I said, you can generate functional information just randomly. And finally, the last term is functional information. That's a term that's first appeared, as far as I can see, in Nature. I think it was 2003, when a biologist said, look, we need to have a special term for the kind of information we're seeing in biological life. It was Jack Szostak. And because he said, normally in information theory, and I know this from engineering, you're just worried about how many bits go from A to B and maybe what the error likelihood is, but you're not at all concerned about what those bits of information say. They could be total gibberish. That's not your concern as an engineer. Your concern is, how can we get it go from A to B with the least amount of contamination? But he said in biology it matters a great deal whether that's gibberish or not. So that's why he suggested we use the word functional in front of information. And a few years later, in 2007, two papers came out. One paper where he was a co-author, Jack Szostak, Hazen was the primary author, showing how we can actually measure, or giving a formula for measuring information, functional information in biology, based on Shannon information, for those of you who know that. That same year, I'd also been working on the problem on how do you identify information encoded in the genome, how do you measure it, and what does it do? And I published a paper that showed how to actually do that for proteins using large multiple sequence alignments, using actual data that's available on the web. Web archives where you can download thousands of sequences for a given protein and do your calculations. So that's the hypothesis. Now, when I'm presenting this at a university campus, it'll be quite a bit different lecture, but there'll be this hypothesis in there. I like to warn people ahead of time that you may see some conclusions arising out of this that you may find disturbing. And the immediate reaction is just to dismiss them. But that's not how we work in science. If you find the conclusions disturbing, you need to falsify this hypothesis. And speaking of that, this hypothesis is testable, it's falsifiable, and it's verifiable. We can test it two different ways. We can test it using genetic algorithms, and we can also test it in the lab. We can knock out a gene, for example, at a particular point and see how long it takes to regenerate that gene, to reactivate it. And then we can knock out that gene at two different points, and we can see how long it takes there, and so on and so forth. So thus far, in every way we've been able to test this hypothesis, it is verified. It stands. So the burden of science is voting in favor of this hypothesis. Not, let me say, this isn't a decision that scientists make. What I should do is rephrase that. So what we're seeing is that when we actually test the hypothesis it's verified, and it is yet to be falsified. So it lives to see another day. You don't really prove anything in science. You just establish that the hypothesis is highly reliable, and if it's really reliable, in biology you can actually call it a law. I'm not quite going that far, but I think it's about the stage that we might have to call it a law. So that hypothesis actually gives us a scientific method to be able to test to see if there was a mind required for an effect. And it's not, you can use this in biology, you can use it in archaeology, forensic science, the Ontario Lotteries Commission could use it to detect lottery fraud. You can use it in a wide variety of applications. So here's the method. Number one, measure the quantity of functional information required to produce the effect. And that can be done using the methods established in the literature. Number two, if the functional information is not statistically significant, then you conclude that intelligence was not required. It doesn't mean there was no intelligence behind the effect. It just means it wasn't required to produce it. You could spell the letter it, and you did that on purpose, but statistically it could happen randomly as well, so you can't claim any stroke of genius in doing that. Number three, if the functional information is statistically significant, then an intelligent mind was required to produce the effect. Very simple and elegant method based on that hypothesis. So what happens when we apply it to proteins, for example? And I chose proteins in my own research because when we're worried about, let's say, how much information is required to produce the simplest life form, there's a lot of room for, or there's getting less in room all the time, because some of you know about Venter's latest research on what's the minimal genome we'd require for life. But let's take something smaller, proteins, just something nice and bite-sized. And so here's what we do when we apply this information, or this method, to REC-A. Now REC-A I chose for two reasons. Number one, it's a universal protein. It's found throughout all of life. And therefore, we can infer from that that it's probably essential to life, REC-A. The second thing is that it's approximately average in length. It's not a really short protein, it's not a really long protein, it's just an average length protein. So what I did here is I downloaded just over 6,600 sequences from PFAM, and then I stripped out all the redundant ones. So if you don't do that, you'll get a higher figure for I, and you run the risk of inflating your results, which I don't want to do. So when all is said and done, you can run it through the program that analyzes thousands of sequences. Typically, it'll be many hundreds of columns and maybe thousands of rows, and it calculates the frequency distribution of each amino acid at each site, and using the formula or the equations published, you can actually come up with a number, the amount of information required to code for REC-A. And that turns out to be a roughly 976 bits of information. So you divide that by 8, you got bytes. You're all familiar with that probably, because you're concerned about that when you fill up the memory on your phone. So what does that mean? Well, what is significant? What's a significant level of information? Because nature could accidentally produce information, so what's significant? Well, that is a difficult number to determine. I'm being generous here at 30 bits. That's generous. Some lab results coming out of the University of Wisconsin where they're knockout genes, double knockout, suggest that it's quite a bit less, maybe 18 bits of information. But again, I don't want to stack the deck. I'd like a high level here. So it's 30 bits. So what that means is if you measure the amount of information required to code for a particular protein, and it turns out to be more than 30 bits of information, then by that method that I proposed there, intelligence was required in order to encode that information into the genome. And that's my conclusion here. Now, you can actually derive probabilities from that equation, because Shannon information at the core is a probability. That works out to obtaining REC-A, let's say through random search, would be about 1 chance in 10 raised to the 294th power. That'd be the chance of finding it in a single search. And I just published a blog a few days ago. What's the upper limit for the number of searches? If you wanted evolution working over a 4 billion year period, 10 to the 30 life forms, fast replication rate, so on and so forth, I came out with 10 to the 45th, which was 2 orders of magnitude higher than an extreme upper limit published by a Scottish scientist, which is 10 to the 43rd. 2 orders of magnitude. I wanted to be more generous. I don't want anybody to fault me for stacking the deck in my favor. So I made some pretty generous assumptions. But still, we're within 2 orders of magnitude. Now, if you're going to search for Rec A randomly in 10 of the 43 trials, but you're searching for a target that occupies only 10 to the minus 294th parts of the sequence space, it's highly unlikely you will find it. You're going to fall short on average by more than 200 and about 250 orders of magnitude roughly. And the problem is that when you're searching for novel protein families, it has to be a random search. Natural selection does not help you. There's no fitness landscape between the protein families. Some excellent papers on that. One of the best I've read just came out in 2014, but that's been known for quite a while. So once you find an existing protein, then natural selection can help fine-tune that protein. It's finding the protein families and you just need to find one sequence for that protein family. Anyone will do, and then you've got it made. But finding them. So what I'm saying here is simply that basically when you go back to the information, the scientific method, when you measure the amount of information required to code for this average protein, it tests strongly positive for a mind behind Rec A. But for life, for the full diversity of life, you need tens of thousands of protein families. For the simplest life form, you need, I think it was 473 protein families. I think, was it 473? Who remembers the exact number? 473. It might be 483, but I thought it was 473. So anyways, the average protein, the simplest life form, therefore, will be in even greater need of an intelligent mind behind it to properly encode the genome of that life form. So that, what you've seen here, let's look at implications for Darwinian theory. For example, microevolution, there's no problem at all because microevolution does not require statistically significant levels of information. In fact, for most of what happens in microevolution, zero information is required. And a lot of times it's actually a loss of information. Very seldom is an increase in information. The Lenski experiment, for example, they had a 4.5 bit increase in information when a promoter region was duplicated, dropped in front of this inactive gene, and then the gene was fine-tuned with 2 mutations. 4.5 bits as you can see, is quite a bit below this being statistically significant. It's insignificant, basically. So for microevolution, no problem. Natural selection works, fine-tunes everything else. It's the macroevolutionary problem where you have to start generating completely novel protein families. Without a mind, you're in trouble because the scientific method that I use here indicates that a mind is required. And as I would say to a secular university audience, I realize that some of you may find this disturbing. And if so, then the way out is to falsify that hypothesis that you saw. And just between you and me, good luck with that, because there's an awful lot stacking up in favor of it. So what am I saying here? Yeah, Darwinian hypothesis requires natural processes to produce significant levels of functional information. That prediction is consistently falsified computationally and in the lab. Therefore, a key premise in Darwinian macroevolution is falsified. In other words, there are certain theories that have fundamental predictions, and then there's peripheral predictions. And if those predictions are falsified, it's not a big deal, just a small tweak. But if it's a fundamental prediction, a key premise, then it's a serious problem with that approach. Now, I was thinking here through in the mind, because it was the Bible and molecular biology, and the first idea in the Bible was that there was a mind behind the origin and diversity of life, which I've just, said there's some scientific grounds for that. Now let's move into the fall of humanity, and see what that effect is in microbiology, for example, in the genome. So we read things like this in the Bible: God saw all that he had made, and behold, it was very good. And then we see in Romans 8, for the creation was subjected to futility, not willingly, but because of him who subjected it, in hope that the creation itself also be set free from its slavery to corruption into the freedom of the glory of the children of God. Now what I note here is, what's with this? Slavery to corruption. How does that compare to very good? And I realize, and I would not, if I'm doing Bible teaching, which I do a lot of as well, I tell the people, don't believe a word I say. Just check it out with the Bible, make sure you think that's what it's saying. And I acknowledge that there's different ways of looking at and arriving at different interpretations of Scripture. I acknowledge that. But I'm just presenting it to you as the way I see it here, or a possibility, and it looks like there's a possibility in Romans 8 that the sin of humanity has actually affected nature itself. That's the possibility that I'm entertaining here. And so do we see any effects of that in science? Excuse me. So, prediction. And in science, it's good to make a prediction. So you got this hypothesis that nature is disintegrating, it's running down. What sort of predictions could we make out of that? And one of them is that we should observe an ongoing deterioration in the genetic information of life. And why did I pick that? Well, I just showed earlier that that has test positive for having a mind behind it. That was something the Creator did, the creative mind did. And therefore, if things are enslaved to corruption, there should be problems developing with the creation. So that's the prediction. So the Darwinian prediction, though, is quite interesting. It's diametrically opposed. In Darwinian theory, you start with zero information, and you slowly build up that information over the eons until it encodes the full diversity of life. So in science, you just love these kind of predictions because it's one thing to have a prediction that doesn't really challenge any other theory. It's quite another when you have two competing theories and both of them make logically incompatible predictions. So if you prove one, it weighs heavily, but that's nice, but it doesn't necessarily get you a whole long ways. But if you can falsify one, that really gives you some good steps to analyze the validity of different theories on. So, observation. Well, let me go back here. First of all, I want to say that all the evidence thus far appears to be falsifying the Darwinian prediction and verifying the biblical prediction. Now, that's just an assertion I just made. Whenever you hear people make assertions, it's not worth the paper it's written on unless there's an argument behind it, so I'm about to show you that. However, let me just say this one thing. Everything we see in reality when it comes to information storage, there is no such thing as a perfect information storage medium. You always get information degradation. Some faster than others. Just heard recently about a possibly new way to store information. It's sort of like a crystalline structure that could preserve information for as long as more than 1 billion years, but it still will deteriorate given radiation effects and so forth. So it should not be surprising. In fact, evolutionary biology is the only area in science where this observed almost law of nature is just simply set aside. The theory is that information actually increases in DNA over time, rather than everywhere else in reality where it decreases. So what is actually going on? A series of interesting papers have been coming out over the last number of years. This was the first one I ran into. But what it suggests is that the whole bacterial world is running down. That there are insertions and deletions in the genome, the DNA, but the net effect is that there's more deletions than insertions. And so what we once thought were a number of different species of bacteria have all descended from one, but not in the right direction. One that had a larger, more complete genome. But the interesting thing about this is that as they lose their DNA, they become increasingly pathogenic, dependent on other sources for nutrients and so forth. And so what I'm pointing out here is a net deletional bias in the bacterial world. And bacteria are one of the things, E. coli, one of the things we've studied the longest. 5 minutes, okay. Next paper, fruit flies. You all heard of fruit flies? We've studied the living daylights out of those things and so if we're going to see a problem, we should be seeing it in the fruit fly, and that's what we see here. In fact, the closing line, these results strongly suggest that the high rate of DNA loss is a general feature of Drosophila, and not a peculiar property of a particular stretch of DNA in a particular species group. So what they're observing in fruit flies is a high rate of DNA loss. The Lenski experiment, for example, that managed to generate 4.5 bits of information, has actually lost massive amounts of information. Reports vary. 8% of its DNA is one report I got from a biology professor. My son was at a conference where he's talking to one of the lab workers. She didn't give a number, but she says our bacteria have lost a lot of DNA. So over 65,000 generations. Things are running down, which is what you would predict. Now, are there any papers to say there's opposite trends observed in any other life forms? No, I've not been able to find anything. The few papers that are out there all are saying in this particular life form or that, we're just seeing a consistent deterioration of the genomes. There's quite a few on human genomes, and they have the same problem. Basically, the problem is this, is that errors are accumulating at a faster rate than they're being corrected. Harmful mutations occur far faster than beneficial mutations. Last section. What about life producing after its own kinds? Okay, again, more room for interpretation here. I'm just going to take a face value look at this, and I realize, again, people have different views on this. But if we take reproduce after our own kind at face value, there should be limits to biological change. That's a scientific prediction you would make from this hypothesis that life has been designed to reproduce after its own kind. So if we play around with life, you will get a lot of variation, but you're going to hit boundaries. You're going to hit genetic limits to change. If Darwinian evolution is responsible for biological life, then there should be no limits to biological change. You might hit a limit here, you back up a little bit here, and sooner or later you'll find the doorways. In every single breeding experiment to date, no exceptions, we always, where these limits, where we try and see how far we can go, one of two things happen. We either have a variation about a mean, which is quite often what we see in bacteria, or say, for example, in plant science, you can reproduce them a little bit not so much ethics involved there, but as far as with animal science, but you keep hitting these genetic boundaries. And now we're beginning to understand why. There's Lenski's long-term evolutionary experiment, 65,000 generations with only a reactivation and fine-tuning of an existing gene, but a much greater loss of DNA. Now, when you have 65,000 generations and nothing happens, you measure evolution in terms of generations, not time. So let's say the average chimp, human type of animal reaches sexual maturity in about 11 years. So that's about, let's say, at least 65,000 years of, what's the number? 2, okay. It has massive implications. In fact, there's another one as well. And that's orphan genes. We're beginning to discover genes that are unique to different organisms that are not found anywhere else in life. For those of you familiar with de novo genes and orphan genes, there's a distinction between the two, so make sure you don't. De novo can be randomly assembled from non-coding stretches of DNA, appears to be non-functional, and leads to intrinsically unstructured. So here's the way one fellow says it. Bacteria, the simplest form of independent life, an ideal for this kind of study with generations that are fast. It says, after 150 years of the science of bacteriology, there is no evidence that one species of bacteria has changed into another. They can degrade, as I've said before, but not change. Now, you look at how many generations, about 1.7 million generations. So for, let's say, something reproduces, reaches maturity in about 10 years, that'd be about 17.5 million years of human history, but we're supposed to have only evolved 3 million years ago, roughly. And so you can see there are massive, massive implications for life forms that maybe take a year to reproduce. So the sheer number of generations, we're not seeing anything happening, but what we are seeing is this verification of this prediction that life should only reproduce within its own kinds. There is a way around that, of course. It's called genetic engineering, but as soon as you got genetic engineering, you got intelligent minds involved in the lab saying, well, let's make this or let's make that. Last slide. This is shocking. I still am taking it with a grain of salt, but there's a new study by astrophysicist Eric Zachrison. Estimates that there are a total of 700 quintillion planets in the universe, but only one that's Earth-like. That's published in the Astrophysical Journal just this year, and using all sorts of different ways to do that. I find that difficult to believe. That's too obvious. Would God make things that obvious, that we're the only planet in the universe capable of supporting life? So I am really, I say, okay, this is a peer-reviewed paper, and it's the latest numbers, but that's an amazing thing. Even if it's even if it's not 1 in 700 quintillion, maybe it's only 1 in 500 billion, that's still highly unusual. Just thought I'd close with that. There's, resources. I write on this, I blog on this, but my blogs are designed to be permanent references with well-referenced links to other things and searchable. That's my blog there, and as I said before, you can download the notes at that location. I have another seminar starting 60 seconds ago.